Hello students, in this particular video I will discuss about the entire syllabus of sericulture. This is a quick revision of the entire syllabus. So what is sericulture? Sericulture is the culture of silk through rearing of silkworm. Now India is the second largest producer of silk as China is the first one, and both China and India together produces almost 60% of the entire silk that is produced worldwide. In India, Karnataka, Andhra Pradesh and Tamil Nadu are the major producers of silk. There are majorly four types of silk: mulberry silk, eri silk, tashar silk and muga silk. These are produced by these silkworms. I have given the scientific name. Mulberry silk is by Bombyx mori, and these are the plants on which they feed on. Bombyx mori feeds on mulberry plants, and the other one they feed on castor plants or bear plant, oak plant, fig plant or Machilus species. This is the plant on which the muga silkworm feeds. These are the types of silk. These are the silkworms that produces this kind of silk, and these are the plants on which they feed. So the entire silkworm or the entire sericulture, it can be divided into mainly three stages. First one is cultivation of food plant, in which the food has been produced. That means all these plants are produced. This comes under the agricultural division because it only deals with the plants. Then comes the rearing of the cocoon, in which the growth of the silkworm is mainly focused. It comes under entomological division. And the third one is reeling of the cocoon or weaving of the cocoon. This is the technical division, in which there is a requirement of machines and factories are there. Even though the factories of most silk sericulture is simpler, we will discuss till this much, till the reeling one, because weaving we don't need. Weaving is actually making cloth from the silk thread. We are not bothered about this weaving process, so we will discuss about the reeling one. Till reeling one. So this entire three stages is divided into three divisions: agricultural, entomological and the technical one. First we'll start with the agricultural division. In the agricultural division, now we will focus mainly on this mulberry silk, which is produced by the Bombyx mori, and they feed on the mulberry plant. So mainly our entire focus will be how the mulberry silk is produced by the Bombyx mori. Now they feed on the mulberry plant. So the cultivation of mulberry plant, this process is called as moriculture. But within the moriculture there are many alternative plants are also produced, but moriculture actually is the cultivation of mulberry plant on which the Bombyx mori feeds on. Typically Bombyx mori, they feed mainly and mostly on mulberry plant and nothing else. Now, for growing this kind of plant, there are certain factors which are required, like the temperature, which has to be 24 degree to 29 degrees Celsius. The rainfall, that has to be 600 to 2500 mm per year. The humidity should be 65 to 80%, and the daylight should be around 5 to 10 hours. The soil should be deep, it should be porous and it should be fertile, and the elevation should be about 300 to 900 meters above the sea level. So these are the factors which is required for this moriculture. Now, how these plants are grown? First of all, they are propagated. How they are propagated? Either by seedling, that means we will put seed in the soil, either through vegetative propagation, that means we'll put some part of the plant on the soil and then it will grow, or else we'll go for this tissue culture, endoplasmic culture, which is done in the lab. And GMP means genetically modified plants, which is obviously done within the lab, so that we can get a better variety of the plant, and those plants do not get many diseases, so that the silkworm that are feeding on them are healthy, obviously. Now they require irrigation. Irrigation means watering of the plants. Manuring, you know what is manuring? That is giving fertilizers and manure so that the plant is good health. Pruning means in proper cutting of the plant so that they do not very big, so that the growth is controlled. And then finally harvesting, because what they do is that we do not put the silkworm directly into the plant. Rather what is done is that harvesting it is done. Harvesting of both the fruit and the leaves. Harvesting of leaves is done, and then those leaves, either only leaves are picked, or is the whole shoot has been cutted, or branches are been cutted, and then these things are being given to the silk worm so that they can feed on it. And the other thing that I said is fruit. Now the fruit of the mulberry plant is used for other purpose, like there are certain other things which are produced by sericulture, not only silk but also the fruit of this mulberry plant are used for making certain medicines. They are used for making jam, jelly and other food products. So that is also one kind of advantage of doing sericulture. Now this plant may get some diseases, like they can get some fungal disease or something like that, which is usually treated with some fungicide or pesticide and all these things. So this is the agricultural part. Now we will move on to the entomological part, entomological division. Since we are talking about this mulberry silk, now mulberry silk is produced by mulberry silk moth. The scientific name is Bombyx mori, and 92% of the silk in India is produced by Bombyx mori. Now the classification of this is phylum Arthropoda, class Insecta, order Lepidoptera, family Bombycidae, genus Bombyx, and species is mori. Now we will discuss about the life cycle of this particular organism. It is a holometabolous insect. Holometabolous insect means any insect that completes metamorphosis in many steps are called as holometabolous. It completes its metamorphosis within 4 stages. What are the 4 stages? These are the 4 stages. Now they have sexual dimorphism in adult pupa and larva. That means the adult pupa and larva of the female one and the male one will look different from each other. The voltinism of silk moth can be univolt, bivolt or multivoltine. What does this mean? It means voltinism actually depends upon the number of brood or generation produced in a year in the life cycle of the silk moth. That is called as voltinism. If it is univolt, then within their life cycle, within 1 year, they will produce only 1 generation. Bivolt means within 1 year, they will produce 2 generations. If it is multivolt, that means they will produce more than 2 generations within a year of their lifespan. Okay, now we will discuss the life cycle in little bit more detail. Now first comes the adult moth. The adult moth, they lives for about 3 to 6 days. They do not feed and they do not fly. These adult moths are usually covered by epidermal scales. They have 3 distinct body parts: head, thorax and abdomen. The female one is usually larger and less active. It is usually larger and less active because it has a huge amount of eggs within its body. And the males are usually smaller and they are usually more active. Now this adult moth will produce eggs. So female adult moths will give rise to or produce 150 to 300 eggs in 1 to 2 days. So there are many number of eggs are produced just in 1 to 2 days. These eggs, they are covered in a gelatinous secretion which is again produced by the female moth for the protection of the eggs. And the eggs are centrolethal. What is centrolethal? Centro means central. The eggs in which the yolk is present at the center are called as centrolethal eggs. Now the eggs can be of 2 types. One can be diaposing eggs or hibernating eggs, and the other one is non-diaposing or non-hibernating eggs. What is the difference? The difference is that the diaposing eggs, they go in diapause or they go in hibernate. Usually they are laid by the temperate races and they can remain dormant for about 4 to 10 months, and usually they hatch in the next spring. And in case of non diapause or non-hibernating eggs, they are mostly laid by the races which are tropical, and they hatch within 9 to 12 days after the eggs has been laid. So therefore, these kind of eggs which are non-hibernating and non-diaposing, they do not require much preservation, because as soon as the eggs are being produced within 9 to 12 days, they will hatch out. But in case of diaposing eggs, they require almost 4 to 10 months of preservation. So they require a huge amount of preservation so that they can be produced, so that they can be hatched within the next spring. Now these eggs, after hatching, will form larvae. Now the larva will first, after hatching, will be first coming out as first instar. Then it will develop into second instar, then into third, then into fourth, and finally into fifth instar. So this is how they keep on developing. Now, when the first instar develops into second instar, the point of the transition in which this happens is called as molting or ecdysis, in which they will shed off their old cuticle. This is the process which takes place when one instar changes into another instar, and the process is called as molting or ecdysis, in which they shed off their old cuticle. In this particular period, they stop feeding entirely. This will happen in every stage, like from 1 to 2, again 2 to 3, 3 to 4, 4 to 5. In every case, they will be molting, from moving from one instar to another instar. Now finally, this fifth instar is also called as the ripe larva, because it is the most mature larva, that it is called as a ripe larva. After they had been ripened, or after they have reached this fifth instar stage, then these larvae are picked up and are moved into mountage. Mountage are certain places or certain basket kind of thing which is made up of bamboo. In those, this ripe larvae are been shifted. Now as soon as the live larvae are shifted within the mountage, usually in one mountage one larva is shifted, they will stop feeding. They will not eat anything. And this instar, this 1, 2, 3, 4, 5, till the development, they feed a lot. The size increases in a very rapid way. But as soon as it reaches in the fifth instar stage, they will completely stop feeding, and whatever they had taken up, they will use up the energy, and they will give out a huge amount of excreta. Usually the color of the excreta is reddish in color, because they contain a huge amount of tryptophan. Later on, this excreta is also collected and is used for making dye or other product, which is also an extra income from the sericulture. Then this larva, they will start producing a waxy material within the mountage. Then this waxy material will sticky material, so it will stick to the mountage, and they will slowly start spinning and will form a cocoon. The thing that happens is that, suppose this is the larva, they will start making cocoon. Now when they are making this cocoon, obviously, and first they'll make the outer, then slowly they comes towards the inner side, and finally makes a innermost layer. This innermost layer is usually very hard, or the last layer. This last layer is called as pilar de layer, and this layer is unreelable, because we cannot reel this part, this inner layer, and therefore this part is usually been thrown away. So and this is what? This is cocoon. This is how a cocoon looks like. Okay, now this one, multivoltine and univoltine and bivoltine. Multivoltine usually takes 21 to 25 days from one instar to become the ripe larva, that is the fifth instar, and univoltine, bivoltine, they take almost 24 to 28 days to develop from first instar to the fifth instar. Now how to differentiate, how to know which cocoon is the female one and which one is the male one. To know this, now the female cocoon are usually heavier and the male cocoon are usually lighter, because female one, the size of the female is usually larger than the male one, therefore the weight difference is also there. They are the female one is heavier and the male one is lighter. Then this larva will develop into pupa inside the cocoon. Now since the pupa is already present within the cocoon, therefore obviously they cannot feed, they cannot do anything. This is the inactive stage in which they just develop into an adult. This process is called as pupation. Now after pupation, whatever cocoon we have gathered, from that 30 to 40% of those cocoons will be kept aside so that they can be later on developed into an adult moth. And about 70 to 60% of the cocoons are used for the silk. So rest, the 60 to 70% of the cocoon will be sent further for the further processing, and this 30 to 40 will be preserved so that from there we can develop and we can get the adult moth, because obviously we need the adult moth. Without the adult moth, we will not get any more eggs. Now how to differentiate, or how to know which cocoons are to preserve, because obviously they need to preserve weight wise. So in that way they understand, for the female one is usually heavier and the male one is lighter, so in approximately in 1:1 ratio they will select this female and male one and they will preserve almost 30 to 40%. Now the pupa, we can also differentiate the pupa by looking at them. The female one, they have no spot on the 9th abdominal segment, but the male one, they have a small spot on the 9th segment. By this we can understand which one is the pupa, which one is the female pupa and which one is the male pupa. So finally this pupa will develop into adult moth. So this is how the cycle goes. Next comes the structure of silk gland of silkworm. Silk gland is actually a modified labial gland which is called as the silk gland. They develop in the 4th or the 5th instar larva, and the location is ventral to the alimentary canal. This is the diagram of the silk gland. These are the parts that I have labeled. Now in this, there is a gland called Philippi's gland or lioness gland. The function of this gland is to secrete waxy material and to lubricate the passage so that the silk which is produced can be easily secreted from the silk gland. Now it has got this anterior part, the middle part and the posterior part. This posterior part, they secrete fibroin. The middle part, they secrete sericin 1, 2 and 3, and it is also a reservoir of fibroin, because this fibroin is produced by this posterior part. They will be secreted from here, they will come here, and in the middle part they will be stored. So this acts as a reservoir of the fibroin and it will secrete sericin. The anterior part has got no such function. It will just collect the sericin and fibroin from the middle part and will pass on. Finally, this Philippi gland will lubricate the passage and therefore the silk will come out of this silk gland. Now what is silk? Silk is a natural protein fiber secreted by silkworm in making their cocoon and collected to make thread and fabric. Now 1 kg of silk is collected from 5,500 to 6,000 cocoon. So this many cocoons are required to produce or to manufacture 1 kg of cocoon. Silk is primarily made up of 2 proteins. One is called fibroin and another one is called sericin and some other products. Now the fibroin and sericin is present in 80 to 20 ratio. That means 80 part will be fibroin and 20 part will be sericin. This fibroin is a fibrous protein and sericin is a gelatinous protein. Therefore, fibroin will form the core of the silk thread and the sericin forms the outer coat. This is secreted by the posterior part and sericin is secreted by the middle part. Fibroin is not soluble in water and sericin is highly soluble in water. They dissolve very easily in water. The fibroin, there is no pigment present. In case of sericin, maybe there is some pigment present. It actually depends upon the race of the silk moth or the type of the silk moth. Other than these two proteins, there are certain other things which are present within silk, like a waxy material. There are certain carbohydrates, there are certain minerals, there are certain coloring agents. All these things are also present within the silk. Now we know exactly how we are getting the silk. Now let us discuss exactly how we are doing the production of it. That is, now we will talk about this one, the reeling of the cocoon. That is the technical division. Now in the technical division, this is called as rearing process. This is known as the rearing process, in which the first thing that has been done is disinfection of cocoon. That is, the cocoons that have been collected are disinfected. Disinfection means to clean them. This cleaning process is either done physically, chemically, or by using radiation. But since the physical process is the easiest, therefore usually they go for the physical one, in which they use either sunlight or steam or hot air to produce, not to produce, but actually to dissolve this sericin protein, which I said is very easily dissolvable. And by doing this, the pupa within the cocoons will die. Usually this is done for the 70 to 60% of the ratio, which is taken up for the rearing process. Now, after the eggs has been hatched, the first thing that is done is brushing, in which the newly hatched larvae are brushed from the eggshells and are moved into the rearing trays. So the eggshells will be removed and the newly fresh larvae will be brushed and will moved into the rearing trays. From there, feeding of the larva. Now the larva will be fed by what? Mulberry leaves, which is harvested from the mulberry plants. Then spacing is done. Why spacing is done? Because in one tray they will not put all the larvae. They will use many trays so that there is sufficient space within it. I have told you that the instar larvae, like first to second to third to fourth, they eat a lot. They take up a lot of mulberry leaves. They eat a lot. They grow a lot. So obviously the size will be very big. Like very big means not that big, but actually they keep on increasing the size. So obviously the space required by each instar larva is more and more and more. So space is required. It is good for the hygiene also, because obviously they will keep on excreting and molting and everything. So bed cleaning is done, because they keep on molting from one instar to another instar. They will excrete. Some of them is definitely going to die, and obviously they might get some disease. Obviously this is tried that there should be no disease or anything, but sometimes some of the larvae might get diseased. In that case, it's recommended to remove those very fast, so that the health of the rest of the unaffected larva is not affected. Then mounting is done, in which the ripe larva, which is the 5th instar stage, will be transferred into the mountage. I just told you how it is done. Then after mountage, harvesting of the cocoon will be harvested, and for after mounting, it takes almost 2 to 4 days for a complete spinning, and so finally after 2 to 4 days we'll get the cocoon. After getting the cocoon, this cocoon are being dried. How they are dried? In the sunlight or by using steam, so that the pupa which is present inside the cocoon dies. so that we can store, because if we do not dry the cocoon and we directly store it, the pupa which is still alive in it will die slowly. If they die slowly, they will start disintegrating, they will start decaying, so the entire cocoon will be damaged or destroyed. Therefore they kill it and then they dry it and then they go for storage. A cocoon can be stored for maximum 6 months, no more than that, because if they store more than that, then they will start losing the quality of the silk. Then they are sorted according to the color, shape, weight and the filament diameter. Filament diameter means the diameter of the silk thread that they are about to get from it. According to this, they separate it, because according to that sorting, they will have income like good quality, moderate quality, bad quality and so on. So for that, the sorting comes after the storage. After that, whatever cocoon that has been sorted according to the quality, then they will boil it and cook it. In the previous boiling and cooking, it was for killing the larva. Now they will cook or boil it so that the sericin protein, which usually dissolves, will be completely dissolved and will be removed. Next, deep flossing and brushing brushing tangle. The floss will be removed so that we can get long threads which is deflossed. Then reeling is done. In case of reeling, the thread will be a single thread and there will be no breakage, a very very long thread. And finally, this raw silk that we are getting will be chemically processed, because still there might be some impurity present in it. So to remove those impurities, finally chemical processing is done, and then again re-reeling is done, and then finally packaging is done, and finally they are marketed. So this is how we get silk, and finally the reeling process is done within the factory. Now, disease management. I'll just discuss some of the diseases that the silk moth might be suffering from. These are the causative agent and these are the symptom. Dermestes beetle. These are the beetles that feed on the silk pupa and they pierce freshly spun cocoon, and therefore the cocoon becomes unreadable. Oozy fly. This is a fly. What they do is that they lay eggs on the Bombyx mori larva, and therefore they get black spots, they lose weight, they stop feeding, and obviously they eventually die. So as soon as they get affected, the best remedy is to remove them or to treat them. Then the disease called Debrina, which is caused by Nosema bombycin. This is a bacterial infection in larva, pupa and adult. And this is a fungal infection caused by Muscardine calcino, caused by Aspergillus flavus. This is a fungus, and due to which there will be mummification of the larva, that is the larva or the pupa will become very hard because of the deposition of calcium oxalate. So this is what happens due to the diseases, and the best treatment that is done is usually they are treated with certain medicines, certain chemicals, or they are entirely removed from the place so that only the healthy ones are present out there. Now for the test. Yes, you people are going to have a test on 4th July at 4 p.m. There will be 30 questions of 30 marks. That means one question for 1 mark. I'll not give any extra question. I will give just 30 questions. So make sure that you are answering all the questions. Now the questions can be of 3 patterns like I have given. Either it will be in the MCQ form, or else it will be in the fill in the blanks form, or it might be a true false form. In this format you will get question. You will just have to use your email ID. So make sure that you are having a valid email ID for this. And the test will start at 4 p.m. and it will end at 4:30 p.m. That means you'll get just 30 minutes, 1 minute for each question. One question for 1 mark. So I'll suggest you all to please put alarm before you are before the exam. I think you should put your alarm at least like 10 minutes before 4 so that you don't miss it. be ready for the exam. Okay, and whatever I have discussed today, all your questions, all the 30 questions will be from the part that I have just discussed. So go through this video, and if you have any problem, then you can ask me.